Okay, good morning. Welcome to our grand rounds. Today I had asked Julio to come speak to us. Julio has a very long CV. He's a very accomplished scientist here. For those of you who don't know him, and many of you in the room have interacted with him on various projects. He's a tenured professor, and he has multiple appointments in hematology, oncology, oncologic sciences, and one of his appointments is in our department, and he really is our head of head and neck translational research. And you know, one of the most frustrating things about being a cancer surgeon is that you provide what you think is a really good surgery to a patient, and you know, all of the traditional things that we look at, like margins, status, are good, and then they suffer recurrence frequently, and that's a very frustrating situation for you and for the patient, primarily, and their family. And so Julio's work centers around why this occurs, and he is really an expert in dormancy of tumor cells, tumor metastasis. And so I asked him to come tell us why this happens to us in a not infrequent basis. So he's going to give us some insight into why these some of the mechanisms around tumor metastasis.
Great. Thank you for the chance to talk to this group. Brett told me I should show some movies, so I brought them with me. So as Brett said, my lab is interested in modeling and understanding the biology of residual disease and metastasis and what happens during periods of minimal residual disease and how we can target the disease there. So my lab is part of a NCI-funded tumor microenvironment network center that has 4 institutions: Einstein, Mount Sinai, the College of Nanoscale Sciences at Albany, and University of Wisconsin-Madison. And we have been working on developing technologies and methods and understanding the mechanisms of dormancy. And we also receive funding through other sources at the NCI focused on this program. We also have relationships with industry and private and state foundations that have, or funding sources that have supported our program. And for those who are not familiar with our research, as I mentioned, we're interested in the biology of DTCs, or disseminated tumor cells. But these are also cells that are left behind in surgery margins, and that's a problem in many cancers. And we're a basic science signaling lab that arrived to this problem by studying how tumor cells decide to grow or not grow based on the balance of specific signaling pathways. And we have focused on understanding how mitogenic signaling and stress signaling are regulated by adhesion receptors, protease receptors, and how these control these very important pathways in cell fate, such as the ERK pathway, the P38 pathway, and the unfolded protein response. How cells integrate these signals to decide if they will enter quiescence and after dissemination, or in response to therapy, for example, if they were senesce and remain in a harmless phenotype for a long time. We also want to understand, and we have studied, how these quiescent cells that remain in the body survive for a long time. We had to tap into different mechanisms, biological mechanisms, to understand this process. So we have studied a lot of adult and embryonic stem cell biology, and how niches that control the quiescence of adult stem cells in our body may be actually instructing these residual cells to adopt this dormant behavior. And we have, because this is a very long-lived phenotype in patients, we thought that maybe epigenetic reprogramming plays a role, and we have also used tools in that field. And of course, we have an intimate link with developmental biology because of these mechanisms being connected to cancer biology, and we're very interested also in immune regulation of these niches. And we have been working on this for quite some time. We published the first paper in 1999 when I was a postdoc, and we have built in layers and layers into this program to understand this biology. The problem of relapse is, I think, the future. If you look at this slide, and I'll jump between data from breast cancer and other cancers to illustrate the points, but I think they apply and will apply to head and neck cancer. So if you look, this is a study compiled by a colleague and friend of mine, Christoph Klein in Germany, and he compiled this data from registries in Germany. And between the end of World War II and the mid-60s, you can see here that most of, and this is all subtypes of breast cancer, most of the patients succumb to disease within 5 years. As we approach the end of the 20th century, now you can see that this curve definitely shifted, and this is probably an accumulation of different procedures that extended the life of these patients. But these patients in total continue to die, and they're just dying later and later and later. And now there are 24-year follow-up in ER-positive breast cancer showing that there's a high risk even at that late time point of relapse. So this is, I think, a trend that is going to impact all cancers. It's already happening. You probably are seeing it with HPV-positive head and neck cancer, with that we have better responses, but late relapse still happens. Other hematological malignancies are being impacted by this late relapse. Multiple myeloma used to be an unmanageable disease. Now they have to deal with relapse after long periods. And like that, I could go through many examples. Prostate cancer, melanoma as well. So this is a complex problem because you have to think of the time frame. You're talking about decades of following up these patients and understanding how these cells can reactivate in target organs after they have disseminated. And as I mentioned, this is relevant for hematological malignancies and other solid cancers. We are trying to build a MRD, or minimal residual disease and recurrence program here at Sinai, and we want to do it multidisciplinary. And I think the problem of HPV positive head and neck cancer may be a very good kind of model of the disease to incorporate into this program. Now, as I mentioned, we know this problem involves the mechanisms regulating adult stem cell biology and metabolic switches. Aging is something that we have to address because, as you can see here, the relapse happens at different time frames, but aging definitely is somehow contributing to this process, and we don't know how that plays a role. We think that there's an opportunity to do a population-based analysis of the host. So many questions come by looking at these Kaplan-Meier curves. If you think that this was a gene signature, for example, and you have patients with the same gene signature dying at 2 years and other patients dying at 18 years, what determines this huge gap in time? So maybe the host and factors in the population and the heterogeneity of the human population, copy number variation, SNPs, that regulate host factors that control the immune system, that control these adult stem cell niches that we have been studying. Maybe that's a place that we need to look to understand how the host is controlling the stochastic rate of relapse that we see over time. As I mentioned, we're very interested in understanding how epigenetic regulation of gene expression also controls this late relapse. And what is good is that this research that we have done has identified markers that tell us if a cancer cell that is left behind is dormant or not. And we have identified also drug targets that we can use for therapeutics. So now this looks at the problem from a different angle. Here we look at the kind of the history of a tumor in a patient over time. And we've always thought of metastasis as cells originating from cells that come from what a pathologist tells us it's an invasive cancer, right? And that has led to a lot of research that has led to model how we can identify the most aggressive variants, because if we find the most aggressive variants and we stop them, then we would control metastasis. However, that has yielded very few therapies to target these red lines here, which are the metastases that can appear, as I mentioned, decades later. So if you look at this tumor mass, surgeons usually continue to be our first line of defense and eliminating this primary tumor mass. And then you have these long periods of minimal residual disease where the cells are left behind in surgery margins, lymph nodes that were deemed not positive, and other target organs that we have no good way of knowing if they have residual cells, except for a few exceptions that I'll mention. And then the next time we treat these patients is when they become symptomatic. Unfortunately, by the time you detect a micromet with MRI, it's about 2 to the 11 cells in that lesion. So this is, for us in the biology that we're studying, this is too late. So our focus has been to try to understand the biology of this residual disease that is left behind, and how to target it at that time and prevent metastasis rather than trying to treat the metastasis. Now, as I mentioned, the dogma has been always focused on this process of late tumor cells. So tumor cells that are rare populations in the primary tumor that acquire the metastatic capacity. However, data from several groups over the last 15 years has shown that in many cancers, including pancreatic cancer, melanoma, breast cancer, and I know a group in the University of Colorado that is working on head and neck cancer also, has shown that, and this is clinical evidence, that cells can leave from the primary site even before they acquire what a pathologist would tell you is an invasive characteristic. And this is termed early dissemination. And we think this is important because if the cells are leaving very early, first, this may be a great source of dormant cells that are left around in the body for a long time, because these cells are yet not genetically very evolved, and they may still be able to respond to cues from the host that control their growth capacity. Now, evidence from clinical studies looking as an aggregate to the cells that are left behind after surgery have shown that you can find them in the lymph nodes, obviously in places that you can obtain samples with minimal perturbation of the patient. So lymph nodes and bone marrow aspirates, as well as blood draws, show that you can find residual cells in these sites. And most of the time, actually like 95% of the time, these cells are completely negative for proliferation markers. Now, this is quite remarkable because they came from a tumor that was growing aggressively, and somehow is counterintuitive to think that a cancer cell with all these genetic alterations somehow enters a growth arrest. And how that happens is something that we have been studying. Okay, so what we have found, and what the evidence from the clinic is telling us as the common denominator, is that these disseminated tumor cells and cells in surgery margins enter a state of quiescence, which is by definition a reversible growth arrest. But it's a very profound growth arrest, and it has very strong survival signals that allow these cells to persist over time in the target organs. Now, we really didn't know how these cells that are coming from these premalignant lesions could actually complete all the steps of the metastatic cascade, because by definition, they shouldn't do it. So we have spent a lot of time trying to understand this. Unfortunately, I won't be able to show you all of the data about this mechanism. It's mostly related to breast cancer in our studies. But we are now trying to take a more comprehensive analysis of metastasis by understanding how these early cells and these late cells that in many patients accumulate over time in the target organs cooperate to form metastasis. So today I'll tell you show you data about how we have found that tumor cells that reprogram into this Ki67 negative state when they disseminate integrate retinoic acid signaling and other pathways to regulate this state of dormancy. So a major problem that as researchers we have is that it's very hard to find models that replicate what you see in humans in mouse models, for example. But there are some commonalities that can be extrapolated and that can be explored in mouse and that reflect scenarios in humans. And one thing that is quite common in humans is that the lung is a permissive site for metastasis, while the bone marrow is restrictive. Now, obviously, there are cancers that preferentially metastasize to the bone, but an important distinction is that they can do it after very, very long periods of latency or dormancy, suggesting that for a long time, cells that reside in the bone may be somehow controlled by the microenvironment and preventing their expansion. There are other cases where you find cells in the bone marrow, but they never grow there. Head and neck cancer is a very good example. In about 20 to 30% of the patients, you can find bone marrow disseminated tumor cells, but the rate of bone mets, at least in HPV negative head and neck cancer, is very rare. Gastric cancer, melanoma, and other cancers as well show a similar pattern. You get liver mets, you get lung mets, but the bone marrow DTCs rarely grow. In mice, this is exactly the same. There's no mouse model that spontaneously gives you bone metastasis, but you can find the cells in that microenvironment. However, all mouse models almost invariably give you lung metastasis. So when we kind of looked at this data, we said, well, this may give us some information of what is happening in the bone microenvironment that can take very aggressive cells and somehow reprogram them into this benign or dormant phenotype. So we have published a number of papers over the years where we have explored what is contained in the microenvironment of the bone marrow that can take a malignant cell with all these genetic alterations and convert it into a dormant cell. So what we found is that in this paper we published a few years ago, that the bone marrow microenvironment is very rich in this TGF-beta isoform, TGF-beta 2. And when tumor cells that have the right receptors arrive to this microenvironment that is rich in TGF-beta 2, through the type 3 receptor, activate a number of pathways, including the P38 pathway, that they turn off the ERK pathway, which is, as you probably know, a target of much of the targeted therapies that go from any RTK down to MEK inhibitors. And there's also ERK inhibitors now. So TGF-beta 2 turns off the ERK pathway as powerfully as any MEK inhibitor or ERK inhibitor. But the other thing that TGF-beta 2 does is activate P38, which usually the inhibitors don't do. And this, through another canonical TGF-beta signaling pathway, turns on CDK inhibitors and induce this profound growth arrest of these cells in the bone marrow. We also discovered that in the bone marrow, retinoic acid can turn on TGF-beta 2 and can turn on a program of quiescence that is driven by the balance of ERK and P38, where P38 predominates over the ERK pathway. And we also found this orphan receptor of the retinoic acid family that is induced by retinoic acid and changes the epigenome of these cells that reprogram into dormancy and drives a very powerful network of genes that drive this profound growth arrest in these disseminated tumor cells. And this work was also published last year in Nature Communications. And one of the things that we found is that we can use currently available drugs, FDA-approved drugs, and turn on this whole mechanism again by using a combination of very low-dose azacitidine and retinoic acid. So if you use these drugs separately, and I'll show you the data later on, you don't get much of this signature coming back in these malignant cells. So just to kind of put it in perspective, if you were to look at all these genes, they're turned off in lung metastasis and they're turned on in the dormant cells in the bone marrow. And you can turn on these genes again with a combination of AZA and ATRA. And NR2F1 is an orphan receptor of the retinoic acid family, so it has a ligand binding domain. So you can actually use the crystal structure of this receptor and find drugs that could activate it. And we did that, and we have now identified agonists that can specifically turn on this orphan receptor and drive this dormancy program. So when do these dormant tumor cells originate, and where can we find them? So I mentioned that early dissemination is a process that seems to be happening in many different types of cancer. So with our collaborator, Christoph Klein in Germany, we have been studying in breast cancer when these cells originate. And we have been doing genetic analysis of single cells, so whole genome analysis of one cell. Here are 740 cells. This is a cumulative array CGH data for the genome of these cells isolated from primary tumors and compared to disseminated cancer cells in the bone marrow of patients that had metastasis, so M1 state. When you look at this, you find that there's specific gains in green and losses in red that are common between the primary tumor and the metastasis. Here's the chromosome that has HER2 amplification. Here's 8q24, a typical amplification in many cancers, including head and neck. So what this data shows is that they're similar. So when you look at this data, you say that makes sense. They come from the primary tumor. What is so surprising about this? They share the same genetic profile. However, if you now look at the profile of many of these cells that were coming from these primary tumors, but now from the M0 patients, so patients that were treated and at the time of treatment had no metastasis. But you can find these cells in the bone marrow already. When you look at the genetic alterations, they look very different. And in fact, they look less evolved. It looked like they didn't have a chance to evolve at the same pace as the primary tumor. That's one thing. And the other thing that this data suggests, and there are many interpretations to this, is that the cells were able to disseminate before they acquired all these genetic traits that are associated with malignancy. And this tells you, or suggests also, that these very aggressive genomes that we see down here may not be required for the process of dissemination, that the cells can leave without gaining all these genetic alterations. The other concept that comes from this slide is that if these cells leave early and they have less evolved genomes, they can evolve in a separate way or in a parallel way in the ectopic tissues. And when we treat metastasis and we think that they look the same as the primary tumor, they may not. Okay? So, and now there's a lot of genomic studies supporting that metastases are undergoing both linear progression. So what you see here is a linear progression model going from the same genetic alterations in the primary side to the metastatic cells, or a parallel progression where you find cells that evolve in a completely different way. Okay, so that gives you heterogeneity of the metastatic disease, and if you choose a therapy based on what's in the primary tumor, you may be missing a large number of metastases and disseminated cancer cells. So these are the movies. So we have modeled this in breast cancer, and we looked in animals that have HER2 positive breast cancer or ERB2 positive breast cancer before we can detect any tumors in these animals. And we have done exhaustive histology of all these mammary tissues, and we cannot find more than some altered ductal architecture. And already at this time, you can see here the tumor cells are tagged with a cyan fluorescent protein. And what we're doing here is two-photon intravital imaging, where we put a window in the mammary fat pad of these mice, and we can image over time these lesions. So this is a duct, actually, and a neighboring duct in the mammary tissue. And you can see that these very early stages, before we can detect any reasonable tumor mass, the cells are already leaving and are entering in contact with the bloodstream. And you can see it here in red. You can see the blood vessels highlighted by a red fluorescent dye that stays trapped in circulation, and in blue you can see the cells breaking off from the early lesions and entering the stroma and entering contact with the bloodstream. So again, this is happening very, very early. We can gain a lot of information from these movies and do these computational 3D rendering, and we can start to map when the cells interact with the bloodstream. For example, as shown here, this yellow overlap is one of these tumor cells trying to enter the bloodstream. And we can actually document this process of intravasation in these lesions very early in progression. So where the arrow is, you're going to see a cell towards the end of the movie popping in and trying to negotiate its way into the bloodstream through the endothelium, and then finally it goes in. So that's that cell going in there. So this really was eye-opening to us. Things are disseminating very early. And most of these cells are actually not doing much. We were able to track these cells to the target organs, to the bone marrow, to the lymph node, to the lung, and we found that the vast majority of these cells, when you look at proliferation markers such as phospho-Rb or phosphohistone 3, the vast majority of these cells are completely negative for proliferation markers. And even the ones that are positive, what you can see here is the HER2 in red, and the phospho-Rb, which is the proliferation marker in green. Even these cells that are positive are very wimpy in their signals compared to a growing metastasis that you can see here. So for the most part, these cells are kind of innocuous. They don't do much. The problem is that they have the ability to reactivate. So in knowing that they are there and knowing their biology, then we now have an opportunity to target them and try to eliminate them or keep them in that dormant state before they reawaken. So this is the proliferation rate for phosphohistone 3, for example, of these early disseminated cancer cells. So it's very little. But when you look at macrometastasis or micrometastasis, now a large proportion is proliferating. And we find these dormant cells, we have found them even a year after they have reached the organ. So they can remain, and a mouse lives for 2.5 years. So this is half the life of a mouse, which in a human would be a long time, right? So where can you find these cells? So now moving into the head and neck models that we have, which are HPV negative head and neck models, P53 mutant. What we found is that, again, as I mentioned earlier, in the lung, we find this. So what we do is we grow a tumor. They're tagged with a fluorescent protein, green, and we allow the tumor to grow. Then we do surgery, and we track the cells in the different target organs post-surgery to mimic what happens in patients and to avoid any influence of the primary tumor through the process. So what we found is that after surgery, for about 2 weeks, in the lung, not much is happening. The cells seem to undergo some default dormancy phase, and then eventually they start to grow. And as you can see here, this increases in several logs the amount of cells that you can find in the lung. In the bone marrow, we find several logs less cells. We're only looking at 2 long bones in the animals for a matter of processing time. And these very few cells don't change over time. Even when we have a growing metastasis in the lungs, it doesn't seem to feed back to the bone marrow. It kind of remains stable. And you can find these cells because they're GFP tagged, or we can use specific antibodies that detect human vimentin. This is a xenograft, a patient-derived xenograft that came from a lymph node metastasis from head and neck cancer, and they disseminate very efficiently. So if you tag them with GFP, you can find these small clusters of cells. You can also find solitary cells in the lung, and you can also find micromets. And as you can see here, similar to the early disseminated cells, even these cells that come from a very aggressive tumor, when they're in this solitary cell state, they're for the most part negative for proliferation markers. So even cells that have all these genetic alterations, somehow when they're solitary, they decide that it's more convenient to stay in this growth arrested phase. Once you find clusters that surpass 20 cells, then a proliferation starts and it's pretty much unstoppable. In the bone marrow, it's even more remarkable. We find these cytokeratin positive disseminated tumor cells that are positive for P27, the cell cycle inhibitor, but they're negative for proliferation marker. You can see here that almost 80% of the cells that we detected are positive for P27, and none are positive for phosphohistone 3. So a very robust phenotype divergence between lung disseminated tumor cells and bone marrow. So this tells you that you can model dormancy in animals with patient-derived models, and you can find these cells and you can interrogate their phenotype. Now, something that we have discussed with Brett actually in the past is that although you have a growing, increasing burden of cells in the lung, this is just coming from some metastases. It's just grow like crazy, and that really dictates the burden that you see. But you can have a growing metastasis, and neighboring that growing metastasis, you can have thousands of cells that are not doing much. So if you treat oligometastasis, for example, and you remove them by surgery, you have a lot of cells left behind that you just don't see. So that is another biology that we can try to target. If we had therapies that target these dormant cells that are left behind, even in organs that will develop metastasis, we could target. So we have done a lot of signaling analysis of these cells that are recovered from the bone marrow. And to make a long story short, they turn off the ERK pathway as shown here, and they turn on the P38 pathway as shown here. And after looking at a lot of different variants of dormant and malignant cells that we have isolated, we found that the majority of these dormant cells have a low ERK to P38 ratio, and that this is driven by the TGF-beta2 pathway. And this is the result of many papers over several years. So we were curious, what is downstream of this balance between the mitogenic and stress pathways that drives this dormancy program? So we wanted to understand the gene programs, and we pharmacologically and genetically altered the P38 pathway to reveal these programs. And this revealed signals that enforce the disseminated tumor cell dormancy program. And this is a complex slide, so I've not expecting you to look at all of this and understand. I don't understand it. And but what I can tell you is that there are some common denominators. So this is a transcription factor network gene. In squares, you have the transcription factors that drive gene programs, and in circles, you have the genes that are regulated by those transcription factors. That means that's what the arrows tell you that this transcription factor NR2F1 binds to the promoter of all these genes, and it turns them on if they're orange, or turns them off if they're blue. And what we found was this very interesting pattern. When dormant cells enter this dormancy state, they turn off this MYC node. MYC is an important oncogene, so it makes sense that they turn it off. They turn off the NF-kappa B pathway. That makes sense also. But what was quite surprising is that they turn on these differentiation pathways. They turn on the retinoic acid receptor beta pathway and NR2F1, which is a lineage commitment regulator in neural crest cells. It commits the progenitors to a certain state that then allows them to differentiate into very specific neurons. And it also regulates similar processes in other tissues. So this was telling us that, and actually these two genes, RAR beta and NR2F1, are the most highly connected two nodes in this whole network. So kind of everything converges onto these nodes. So this told us that this may be important. And then when we looked at other genes regulated in this network, we found a bunch of genes that are turned on that are all in the retinoic acid family of gene regulation. So we explored this a little bit more, and we wondered if it was important and what was this NR2F1 gene doing. So NR2F1 is a coactivator of the retinoic acid receptor alpha. It also regulates the function of estrogen receptor alpha. As I mentioned, it's a lineage commitment regulator in neural progenitors, and it restricts induced pluripotent stem cell reprogramming. So if you want to make iPS to then generate stem cells and derive different tissues, if you block NR2F1, you can make this much more efficiently. So it's a barrier to pluripotency, if you want. What we found is when we looked at human samples, we did a study in breast cancer with about 1,500 patients. We found that this gene was frequently turned on, along with a lot of these genes in that network that I showed you that we used as a signature. They were more frequently turned on in the ER-positive luminal breast cancer type than in the low dormancy score or ER-negative, triple-negative breast cancer type. Now, this is interesting because ER-positive is the subtype of breast cancer that recurs very late versus the ER-negative, which most of them recur in the first 5 years. So then we used a signature that contained NR2F1 and the other genes, and we performed a Kaplan-Meier curve analysis by looking at the signature in primary tumors. And we asked, if a tumor has an enrichment for NR2F1, let's say, does it tell us anything about metastasis-free intervals? That's the best information we can get about dormancy, how long between surgery and a metastasis event. And what we found is that if the tumors, the primary tumors, already contained this red signature that you see here, they tended to recur later than if they didn't have the signature. So if they had a low enrichment for the signature or a low dormancy score, they recurred pretty much within the first 5 years, like a triple-negative breast cancer. However, they had this signature, they really went kind of slowly recurring over time, suggesting that perhaps these cells are kept in a dormant state by the signature after they disseminate. So in head and neck cancer, NR2F1 is usually present in the benign adjacent tissue. This is immunohistochemistry done with samples that we obtained here at Sinai. This is our malignant HEP3 model, which is our PDX. And the ones that are growing and metastatic, they don't express NR2F1, but the ones that reprogram into dormancy, they turn on this NR2F1 gene. Now, if we look at primary tumors, recurrences, and lymph node mets, in general, this gene is turned off completely. So that told us that somehow, in order for tumors to grow, they have to turn off this dormancy gene. What we found, and I'll show you, is that when the cells disseminate and they reprogram into dormancy, they turn on this gene again. So it's really an epigenetic regulation. So we found that specific epigenetic histone marks silence this gene in growing tumors. But those tumors that still express to some extent NR2F1 usually are associated with node-negative status and delayed recurrence. And this is from databases. And this is all head and neck data. So this is pretty much what you see when you look at, in general, the tumors. The frequency of expression by immunohistochemistry is downregulated compared to benign adjacent tissue. As I mentioned, if we now look at cells that we recover from the bone marrow or dormant cells that we find in the lung, they turn on NR2F1, as you can see here in red. This is gene expression data, and they turn on all these retinoic acid regulated genes, while the proliferative metastatic cells don't express those genes. We can detect the protein upregulated in these dormant cells compared to aggressive variants of head and neck cancer. And in other models of dormancy, we also find, for example, these 4007 breast cancer cells that have a dormant phenotype, they also turn on NR2F1. And in the bone marrow-derived disseminated tumor cells that we isolate from our mouse models, NR2F1 is turned on, while in the lung and in the primary tumor is turned off. So this tells you that coming from the same growing primary tumor, those cells that arrive to one microenvironment turn on these dormancy genes, while if they go to a site where they grow, they turn it off or keep it off. So we did some experiments to understand if perhaps in surgery margins, this gene is also turned on. We thought this was relevant for head and neck cancer. So we allowed tumors to grow, and then we did surgery. And we're pretty good because we don't get a lot of recurrences, right? We got these two here. And they don't grow very fast. So when you inspect what is left behind in these, so we took the animals that don't have any evidence of recurrence, and we sectioned all the wound. So it's easy in the mice because they're big. So we sectioned the whole wound, and we looked for these solitary cells that were staining positive for this human-specific vimentin, and we found them. We found even 1 centimeter away from the margin, from the surgery margin, we could find these cells. So they were migrating also kind of laterally in the skin. And frequently, when these cells were solitary, they expressed NR2F1, this dormancy regulating gene, and SOX9, which is a target gene of NR2F1 and also regulates dormancy. However, if they were proliferating, or in these recurrences that you see here, you can find now that they start to proliferate and they turn off NR2F1, even small cell clusters. And actually, we followed. We found that when we found clusters of cells that were turning off NR2F1, usually a little further away, we could find some larger clusters that were already proliferating. We quantified this, and you find that frequently when you find these residual tumor cells on surgery margins, they are upregulating NR2F1, SOX9, and they're turning off the proliferation pathways. So here we didn't do anything. We just did surgery, and we looked what was left behind. And we could find these cells that were turning on spontaneously these dormancy pathways. So now you can imagine, you have these residual cells. If we were able to kill these cells, we could extend this green line over a very long period of time if we were able to eliminate them completely. Or we could also find ways to keep these cells from reactivating and also extend this blue line. We looked also in the lung of these mice, and we found that within those 2 weeks that I told you that there's kind of a default dormancy phase, these solitary cells start to turn on these dormancy pathways, again, without doing any kind of treatment, just following solitary cells. And this is what a positive cell looks for in the lung for NR2F1, and this is what a cell we call negative for NR2F1. And again, we use vimentin for this detection. And this was work done by 2 postdocs in the lab that have left a few years ago. And what you can see here is that at day 5 after surgery, compared to the expression of the primary tumor, you find an upregulation of NR2F1, and even at day 10 this persists. And other genes that regulate dormancy, such as Dept and P27, are also upregulated spontaneously in these solitary cells. If you look at a growing metastasis in the same lung, they turn off the pathway. So they go through this, they go low from the primary tumor, they turn it on when they become dormant, and then when they switch off dormancy, they turn off this gene. We saw the same in breast cancer. We used quite a cool system, and I wanted to show it to you. So we have a mice that have a tumor that has this fluorescent protein called Dendra2. Now you can beam this fluorescent protein with a 405 nanometer laser or a powerful lamp, and the whole protein will go from green to red. And then if the cell divides, because it's a post-translational modification, so there's no new gene made, the red is diluted. So you can use it as a label retention. If the cells don't divide, they retain the red label. So what we did is we tracked these cells. So the green cells, you can consider them proliferative, and the red cells, you can consider them dormant. So we photoconverted the primary tumor, we allowed the cells to leave, and we checked very early how fast NR2F1 was being turned on. So here you can see in the lung what we call a label retaining cell, because it's green and red, because the the cell was photoconverted in the primary tumor, it arrived to the lung and it retains red label. That means that it didn't dilute it by cell division. And here you have a cell that underwent division and doesn't have the red label. Now, what we found is that the majority of these cells, so we find this is the proportion of red cells that we find in the lung a week after the cells disseminated. So a large proportion of cells are not proliferating immediately as they arrive to the lung. And there's a proportion of these that are NR2F1 positive. Now, if we ask how many of these red cells are also NR2F1 positive, we found that about 75% of the cells that are not dividing are turning on this gene. And almost none of the cells that don't have label retention have expression of NR2F1. So this is, in another model, we validated that this gene may be a very good indicator or biomarker of what a dormant cell is. And that's why we're doing all these detailed studies. So then we wondered, is this gene important for a recurrence? So we generated a system where we turn on an shRNA mirror, which will knock down the gene. It will turn off the gene at will when we add doxycycline, and we can turn on and off the expression of the gene. So here are the controls showing that we can turn off the gene at will. And we did this system because, again, we want to monitor residual disease. So what we did is we allowed the tumors to grow in one group and in the other group as well. And then these lines that drop here and drop here, that's the surgery that we did on the primary tumor. And then in a group of animals, we didn't do anything. We didn't add doxycycline. So these are the animals that I showed you before that had high expression of NR2F1 in the surgery margins. And then in another group, we added the doxycycline to turn on this knockdown, and we downregulated the NR2F1 transcription factor. And in the animals in which we downregulated the transcription factor, we doubled the number of recurrences. And when these recurrences grew, they came back with a vengeance. So the cells that I showed you that had NR2F1 that were left behind sometimes a centimeter away from the surgery margin were using NR2F1 to remain dormant, because when we turn off the gene, they come back. And if we look at the lung and the spleen is a site where we never get metastasis in this model. But if we turn off this gene, now we start to detect a lot of cells proliferating aggressively. And in the lung, we can kind of increase by several logs the burden of metastatic disease, indicating that we were awakening these cells by eliminating NR2F1. So this told us that we can detect the gene and that the gene is functionally linked to this dormant state in those target organs. So can we find these cells and these genes in patients? And what do they look like? So this is data obtained from head and neck and esophageal cancer done in collaboration with a group in Germany. The Germans have been very good at creating clinical programs where they obtain bone marrow aspirates for almost any cancer that you can imagine. And they have used this to monitor residual disease post-surgery and to do genetic analysis of the residual disease. So that's why we have collaborated with these groups very actively. And what you can see here is disseminated tumor cells, the gene expression, the genome gains and losses. Again, now the gains are in yellow and the losses are in blue, from lymph node disseminated tumor cells in these patients and from bone marrow disseminated tumor cells. And what was quite remarkable is that the lymph node, where we all know metastases usually grow in head and neck cancer and also in esophageal cancer, had a higher proportion of aberrant genomes than cells obtained from the bone marrow. And as I mentioned, in these two cancers as well, the lymph node and the lung are permissive sites for metastasis, and the bone marrow is usually restricted. So in the same patients, coming from obviously the same primary lesion, you can find that different sites give you different levels of evolution for those cells. So in the bone marrow, somehow the evolution of the cells is suppressed, and they have less genetic alterations than in the lymph node. So this proved in part our concept that the bone marrow is suppressive, and that you somehow can prevent the evolution of these cells by changing the microenvironment. Then we did work with Christoph Klein in breast cancer, and Christoph has a program in prostate, prostate breast cancer, and melanoma for the analysis of residual disease. And I asked him, can we look at DTCs from these patients? And we looked at each column that you see here is a gene expression profile of cells from one cell from patients that were in M1 state or M0 state. So here the yellow cells are the M1-derived disseminated tumor cells, and the purple are the M0 DTCs. And what you can see is that if you look at the cells that come from M1 patients, they look very tight in their gene expression profile compared to the cells that come from the M0 state. So this is something that we see also in the genetic profiles. In M0 state, when the patients don't have metastasis, it looks like there's a lot of different genomes going on, like alterations. There's huge heterogeneity. And when a patient has metastasis, they kind of funnel into a specific signature. So there's a lot of probability of different possibilities that the cells are exploring to reactivate. Now, what was interesting here is that, for example, here is a couple of cells that came from an M0 patient. So this patient was told you don't have metastasis. But when they looked at the cells, they looked a lot like the M1-derived DTCs. So if you imagine that you had enough of these signatures, you could perhaps start to predict when a cell in a patient that doesn't have metastasis is going to reactivate and give you a metastasis. Okay? That's one kind of output of this data. The other one is that these cells, this yellow cell here and this yellow cell here, came obviously from an M1 patient. So this patient had metastasis. But when you look at the gene expression profile of these cells, they look like the cells that come from M0, so are potentially dormant cells. So this now, again, further supports the notion that even a patient that has metastasis still has a lot of dormant cells around. And if you may remove a metastasis by surgery or use anti-proliferative therapies, you may have a lot of dormant cells that are still blowing through the treatment. So I asked Christoph, can you use the dormancy signature that we identified, this P38 enriched signature, and tell me how it separates these cells? Does it tell us anything about these cells? So he sent me this chart, and he said, this is pretty amazing. He says, your signature identifies exactly the same cells. The cells that came from M0 are enriched in your dormancy signature, and the cells that are coming from an M1 patient don't have your dormancy signature. So somehow your dormancy signature is pinpointing dormant cells for the first time. And now we have a mechanism, he says. We have P38, we have NR2F1. So this, for me, it's quite remarkable and promising because for the first time, we can put our finger on what these cells look like and what they may be doing. We did the same in prostate cancer with the University of Washington. They have a wonderful program there where they do bone marrow aspirates also, and they have a rapid autopsy program so we can get metastasis also from these patients. So here we analyzed about 7 cells from patients that were with no evidence of disease for up to 18 years, and then cells obtained from patients that had metastatic disease. And how we isolate these cells, in the case of Christoph as well, we isolate EpCAM-positive cells from the bone marrow, so these is an epithelial marker in a mesenchymal compartment, so you're not taking by mistake bone marrow cells. And also the genetic analysis and the gene expression profiles validate that. But what we found again is that if you look at a number of cells from advanced metastatic patients versus these NED patients, the signatures look very different. However, a number of these cells that came from the patients that already had metastasis look like the cells that came from NED, similar to what I showed you in breast cancer. So when a patient has metastasis, it has a mixture of cells that look like this, red, and these that look like the NED. So what did we learn from these signatures? So this was done, when I got involved in this project, it was pretty advanced, and they got in touch with me because they said, you know, every time we look at this signature in blue here, or these cells that look like these cells here, the thing that comes up all the time is the P38 pathway. And we know you have published this for now almost 18 years on this, so clearly this pathway is important, and it's coming up in these cells that are isolated from patients that have been free of disease for up to 18 years. So we went back and looked at the whole signature, and then we got involved in the study, and we found that all these genes that are turned on in our dormancy signature are turned on in prostate cancer DTCs as well. So this is an independent validation of our signature. And we found that in general, the genes that are turned on in our dormant cells, actually these come from head and neck cancer model, were also turned on in the cells that came from NED patients versus advanced. So we think this is important because the only way we can use new therapies to treat dormant diseases if we also have a way of monitoring it. And you know, if you want to give a new therapy, you have a way of knowing if it's working, right? So having the markers is important. So can we restore these NR2F1-driven dormancy programs? So as I mentioned, retinoic acid was a main theme that came out of the signature, right? These cells on their own turn on, I only have a few slides left, turn on this dormancy signature. So what we found is, as you know, there have been a lot of clinical trials in head and neck with retinoic acid, and you can suppress growth with retinoic acid in these squamous cell carcinoma tumors. And what we found is that retinoic acid on its own could turn on NR2F1, could suppress proliferation, and could give us this whole signature that we had revealed in our studies. However, this was transient. The moment you stop treatment with retinoic acid, the tumors would go back to growing aggressively, and they would kill the mice. So what we came up is with a system. We knew that NR2F1 was epigenetically silenced, and we could re-express it by using low-dose azacitidine. So what we came up is with a protocol where we restore expression of NR2F1 with azacitidine, and then we treat with retinoic acid. And when we did this combination of azacitidine and retinoic acid, we found that we could maintain the expression of these dormancy genes, even if we washed out the drugs. So it was a true reprogramming. We treated the cells, we removed the drugs, and the cells continued to express these dormancy genes, and they didn't proliferate. And they didn't proliferate for up to a month. Actually, this is 2-week data that I'm showing you, but now we have data tracking them for more than a month. They retain label with several methods that we use, telling us that they are quiescent. So these are different measures that we use to know if they are quiescent. And as I mentioned, if you do this reprogramming with retinoic acid or with a specific agonist for the retinoic acid receptor alpha, you can induce this dormant phenotype, and it's sustained. This is all happening after only 4 days of treating with these two drugs. The cells reprogram and continue to remain dormant. So now we're exploring ways to optimize this therapy, and we're using specific agonists that target only some of the receptors of the retinoic acid family combined with azacitidine. And we're finding that genes that are linked to differentiation of squamous cell carcinoma can be turned on with a combination of AZA with specific agonists. Some of them are in the clinic already, so there's a trial that we could design around these. And they turn on several of the dormancy cues. And just as my last slide, we have also identified agonists that can turn on NR2F1. We actually got a venture capital fund to give us a grant to further explore these compounds to develop a company, because now we have drugs that can specifically address the same gene that is the marker. So we now have a potential drug to turn on this dormancy program in these residual tumor cells. And this is some of the data that supports that the agonist is working. So our hope, and this is my last slide, is that we will have, and I couldn't show you some of the data because it's unpublished, so I had to remove it. But if we had a way of extending this blue line, it would be a nice therapeutic option. And we have found drugs that can turn on this dormancy program. We have drugs that, if a cell already is dormant, can maintain the dormancy program. And now we have a new class of drugs that can kill these dormant cells. We have identified a few pathways that allow only these quiescent cells to survive. So we can go with small molecule inhibitors of some of these pathways and eradicate these cells. And we have a lot of data on this. Unfortunately, I cannot show it to you. So this has led to the incorporation between MSIP and hopefully some venture capital funds that are going to invest into this company called HyberCell that is going to try to bring this new modality of treating dormant disease into the clinic through clinical trials. And I hope head and neck cancer will be one of those. So this is the lab. Some of the people were not in the photo, so they photoshopped themselves in as hanging heads or jumping. And some people have left already, and these are collaborators. We have a very strong collaboration with Emily Bernstein here at Sinai, with Eduardo Farias, who is he's a retinoic acid signaling expert, and Emily is an epigenetic regulation expert. And we have benefited a lot from these interactions. And these are the funding agencies. Thank you.
Great. Thanks a lot, Julio. Does anyone have any questions? Yeah. What are the effects in the rest of the body on these drugs that promote dormancy? I mean, obviously they must have some other function.
Yeah. So
Yeah, repeat it because we're
I'll repeat the question. So what is the effect of the drugs that would be used to treat dormancy? So In normal cells. So retinoic acid signaling is required for differentiation. So you would imagine that perhaps you would drive a little harder differentiation of some progenitors in the treatment. And this actually happened. When patients were treated with retinoic acid, you get granulocytic differentiation accelerated. And some of the side effects of that is that you get edema in the lung because you get a flooding of the lungs with neutrophils. So, but one of the good things about 25 or 30 years of retinoic acid studies in the clinic is that people know how to handle the side effects. The same with azacitidine. It's been used. Lou Silverman was the first one to use it, and other people are using it at lower doses than Lou used to. So a lot of when I presented this for a clinical trial, the doctors were quite comfortable with it because we know how what to do with these. So we have two other the agonists that for NR2F1 we don't know because they're new. We're testing them. Again, it's a differentiation lineage commitment regulator that works primarily in tissue differentiation and during embryogenesis. So our prediction is that in adult tissues, it may again drive maybe differentiation from stem cells or progenitors. The concept of having a therapy that is a reprogramming therapy is what we like about this because we may not have to give it the treatment continuously. That we may have to do it kind of in a metronomic way to just keep the program going in pulses. So one of the other drugs that we're studying is a biologic that regulates bone morphogenesis, and it's a very strong inducer of dormancy. So we anticipate perhaps some effects in the bone in terms of having maybe more calcification when we give these drugs. The other drug that we have that kills these dormant cells, these are two drugs that came from Eli Lilly that are top grade, and they went through all the PD and pharmacokinetics. They're all done. And we did toxicity studies, and one of these drugs affects a pathway that also is very important in pancreas. So it's important in the cells that produce insulin. And we thought that would be a major toxicity. And at the doses that we're giving this drug, we have a very nice therapeutic window where we target the dormant cells, where we actually have effects on metastasis that are very dramatic, but we don't see any effects on insulin levels, glucose, or any weight loss in the animals. And this was done over a range of doses. So we think that at least in the doses that we have, if we match the same dose in the patient, it may be safe. And may not if it's a drug that kills these dormant cells, and we were to use it in the adjuvant setting, one would hope if you can monitor these cells through blood draws and CTCs or a bone marrow aspirate, that at one point you would stop treating if you eradicate the cells. So, but these are things that we have to test.
So, I just have one question. So, the traditional head and neck cancer HPV negative cell has mutant p53. With HPV-related cancer is wild type p53, but the p53 gene is suppressed by the virus. So if cells if the viral oncogenes are integrated and these cells are now disseminating around the lungs and bone marrow, would that restrictive or permissive environment still be valid? Because
absolutely,
because the virus is there, but that's really going to drive what happens to those cells, right?
So the question is, if in HPV positive you still have a functional p53, but it's suppressed by the viral protein, and what would happen when the cells disseminate? So we don't know. There's definitely not a dominance of the genetic or viral genome because the patients are free of disease for a long time. So something is taking over that suppresses these mechanisms. So the whole dormancy program that I explained is also linked to p53 activation. So these dormant cells in our models have a mutant p53, but interestingly, it has a mutation that allows p53 to induce p21, but it doesn't induce apoptosis. So when the p53 comes back on in this reprogramming, it drives arrest, but it doesn't kill the cell. So we imagine that perhaps in HPV, and this is something that we would have to study, that perhaps there is, because the cells are stressed, maybe the viral oncogene is not produced in the same levels, and you have some reactivation of p53, or some of the other pathways that drive growth arrest can override the lack of p53, and there's some compensation. But clearly that is completely fertile ground. Nobody has ever looked at these questions in a viral, genome-driven cancer.